Good afternoon, everyone, and thank you for joining us for today's webinar. My name is Josh Vickers, and I'm with RTI International's Forensic Technology Center of Excellence, and I will be your host and moderator today. Today's webinar, we do have 3 presenters and 2 presentations. They'll be presenting on estimating blowfly age and reducing error in post-mortem interval cases. Our first presenter to introduce is Dr. Aaron Tyrone. He graduated from UC Davis in 2001 with a degree in genetics and stayed at UC Davis to work in the section of evolution and ecology on Drosophila research projects until 2003. He then moved to Michigan State University, where he obtained his PhD in zoology in 2007, working in the lab of David Ferran. His PhD on the functional and quantitative genetics of Lucilia serracata development was partially funded by an NIJ grant that he wrote with his advisor. He then did a postdoc in molecular and computational biology at the University of Southern California until 2009, when he moved to Texas A&M University to start as a faculty member in the Department of Entomology, teaching entomology and forensic and investigative sciences courses. He has authored several book chapters related to forensic entomology and decomposition ecology, and edited a book on the evolution and ecology of carrion decomposition. His lab studies how insects and microbes can provide information regarding timelines in death investigations through an understanding of their evolution, ecology, and functional biology. Our next presenter of the day is Dr. Christine Picard. She received her Bachelor's of Science degree in Biology and Chemistry from the University of New Brunswick and her Master's in Chemistry from the University of Toronto. She then received her PhD from West Virginia University in 2010. She completed a short postdoc in Aaron Tyrone's lab at Texas A&M University before accepting a faculty position at IUPUI in 2011. Her research is focused on the genetics and genomics of blowflies, among the most forensically important insects. Dr. Picard's research laboratory has assembled more than 10 fly genomes and continues to work to understand genotype-phenotype correlations in forensically and non-forensically relevant traits. You'll often find Dr. Picard packing up her rotten meat and grabbing her fly net to catch more flies to gain a better understanding of their biology. Our third presenter of the day is Dr. John Goodpaster. He received his Bachelor's of Science degree in Chemistry from Gustavus Adolphus College, and then followed by a Master's in Criminal Justice and a PhD in Chemistry from Michigan State University in 2000. He completed a National Research Council postdoctoral fellowship at the National Institute of Standards and Technology, NIST, at Gaithersburg, Maryland. He's a former forensic chemist with the Bureau of Alcohol, Tobacco, Firearms, and Explosives, and the current director of the Forensic and Investigative Sciences program. His research focus has been mainly in the investigation of fire and explosions. More specifically, Dr. Goodpaster has pioneered new analytical methods for detection of explosives in post-blast residues, discovered innovative methods for the preservation and identification of ignitable liquids, and developed robust statistical methods for their analysis. You'll often find Dr. Goodpaster out in the field with his students and the Indiana State Police Bomb Squad, detonating devices to understand how the chemical signatures of various bomb types are detectable. Our quick disclaimer: the opinions, findings, and conclusions or recommendations expressed in this presentation are those of the presenter and do not necessarily reflect those of the Department of Justice. Any products and manufacturers discussed are presented for informational purposes only and do not constitute product approval or endorsement by the U.S. Department of Justice. And without further ado, I would like to hand things over to our first presenters, Dr. Aaron Tyrone and Ms. Christine Picard.
Howdy, colleagues. I just wanted to thank the National Institute of Justice and RTI and our colleagues at both institutions for their assistance in making this happen. I'd also like to acknowledge another colleague that's worked on these 2 grants that we'll be talking about, Sing-Hui Tse. He's a co-PI working with us. I'd also like to acknowledge our other collaborators, because this is a highly interdisciplinary set of projects. So this is Cliff Spiegelman. He does statistical research and served on a students committee related to this work. This is Spencer Johnson. He also served on a committee and is involved in population and quantitative genetics research. He's been helping us with analyses. David Riley is at Texas A&M in the animal science program, and he does quantitative genetics. Jonathan Parrott and Carl Hellman are both postdocs that have worked on various aspects of microRNA, protein, and mRNA expression and the selection experiment. Ernie did a master's project on part of the selection experiment. Anne and Sarah worked with Christine on various genetic analyses, and then Yi and a student whose picture I cannot find, Shuhua Fu, worked with Sing Hoi on the bioinformatics related to what we're doing, and Satyam has an interest in microRNAs and has worked with us. He worked with us for a year, and you'll see a little bit of data from what he did as well. So there's a large group of people involved in this. I just wanted to acknowledge them up front. But basically, what we're going to talk about today is this idea of flies as biological clocks in death investigations. So the idea is that somebody dies, and the blowflies that we study are common primary colonizers of animal remains, including human remains. So if you find a dead body and you pick up insects from these blowfly species from the remains, you can back calculate, if you have some information, to when those insects were deposited on the remains, and that gives you some information about the timeline related to that death. And this all relies on predictable life cycle. So this is a typical life cycle for a blowfly. They start as eggs. You can see in this case there's several hundred eggs laid on a piece of liver. These guys hatch into first instars, which are these little squiggly guys around what used to be that egg mass. And then they grow and molt a few times. So they molt into second instars, which are a little bigger. Those second instars molt into third instars. You can see these feeding third instars have food in their crops. At some point, the third instars receive a hormonal signal that triggers pupation to initiate, and so you get these post-feeding third instars, and notice this does not have food in the crop. They stop feeding, and they start looking for a place to form these puparia, and inside these puparia, the flies are going to go through metamorphosis, going from these little maggots here to something like an adult. And so this is a temperature-dependent process, but if you have a good estimate of what the temperature was in relation to your discovered body, for instance, you can use things like accumulated degree models to back calculate to when those eggs must have been deposited. All right. So there are several aspects here that I think are important for understanding what we're going to talk about today. So the first component here is each of these stages gets pretty much longer than the next one. So the egg stage and the first instar stage are typically fairly short, as is the second instar. The third instar can be fairly long, and there are some reasons why sometimes you can't tell the difference between a feeding and a post-feeding third instar. And so you can have a large block of developmental time associated with this one development stage. And pupariation typically is something like the last half of development. So again, it's hard to break that into a smaller piece of time unless you can get inside that puparium and know a lot about the structures of the organism or do some molecular work like we're going to talk about today. Another thing that you can do to add precision to your estimates is these flies in these earlier stages grow linearly with respect to age. And so for the first instars and the second instars, there's an age-size correlation. In the third instar, once they stop feeding, they also start shrinking. So again, size is not as useful for this third instar. So again, additional information in this potentially problematic third instar and puparial stage can help add precision to estimates of insect age. So this is a hobby I picked up in Texas. Apart from the concept of riding a horse at a run while trying to shoot a target, this is basically just an exercise in marksmanship. And I kind of think about this when I think about what people are trying to do with forensic predictions of any type. So the idea is you have an unknown sample. In this case, it would be something like an insect specimen collected from remains, and you want to predict something about it. In this case, it would be the age of that insect specimen. And the ideal is you hit right on the mark. So if this is a 7-day-old specimen, I always hit the bullseye, and in this case, this would be 6 to 8 days. There's some variation in how well you can hit the center of that target, but you're pretty close. So you'd say this is a 6 to 8 day old specimen. So that's accuracy with precision. But there's different ways to shoot at a target. Some people are systematically biased in the way they take their shots. And so you may have a biased estimator if you're doing forensic research and predictions. So this might be something like saying, oh, I think this insect specimen is 10 to 12 days old. You're precise, you have a tight time range, but you're off the mark. So if you have good validation work, you might know this is a biased estimator and that you can correct for that bias. But this is a problem that we should consider in forensics. This is usually what happens when I'm riding a horse at a run and trying to shoot a target. You have accuracy, you can hit the target, but you have a lot of variation with respect to how close to your ideal mark you hit. So in this case, maybe this would be a 7-day-old sample that you're predicting is 4 to 11 days. So you're accurate, you're getting in that range that you're reporting the correct answer, but you might not be providing very useful information to the investigation if it's too imprecise. And then this is an issue that you would want to avoid, is inaccuracy and imprecision. But I think one thing to keep in mind is you can have intermediates, and as you go from this kind of accurate and imprecise condition, and you add precision. So we're going to, in the course of this talk about identifying smaller windows of developmental time. You might find out that you're a little bit biased, maybe not biased like that, but maybe you have a bias where your samples are just above the bullseye, for instance. And this is where, if you're adding precision, you might want to be thinking also about accuracy and how you can improve accuracy. And so we'll talk about both of these today and how we can get at this. So one of the big problems when you do a forensic estimate of insect age is that you have to assume a reference data set. That reference data set is basically, usually takes the form of a table that says this insect species takes this long at this temperature to reach this developmental stage. And if you get into the literature, you'll realize that there can be variance on those numbers, even within species. And that can be for a number of reasons, both the environmental conditions that went into producing that reference data set and the genetic conditions. So which specific individuals of that species are you looking at to develop your reference data set? And so if you have an environment that doesn't match your casework conditions, or you have a genotype that doesn't match your casework conditions, you might end up with inaccurate estimates. And we'll talk about this later, but the resolution of this may also allow you to increase precision because you might be able to identify a subpopulation that you're dealing with, which has a restricted distribution of traits than your overall population. And so just to give you an example of this, in my dissertation work, this is from Tyrone et al., 2011. You can see these bar charts here and reaction norms here. So we took 3 different populations of Lucilia sericata, one from Davis, California, one from East Lansing, Michigan, and one from Morgantown, West Virginia, and raised those flies. So we took a bunch of different cohorts from each of these groups and raised them together. This is basically what's called a common garden experiment, essentially. So if you put them in the incubator together and let these cohorts grow at 20 Celsius, they all grow about the same development rate. Accumulated degree hours is basically a way of estimating age, accounting for the fact that insect development is temperature dependent. However, if you go to 33.5 Celsius and you raise the same groups of cohorts, 2 of these strains, the Michigan and the West Virginia strain, develop much faster than this California strain, which systematically develops slower. So this is an example of a genotype that in at least one condition develops slower than other members of the same species. There are other examples of this. Gallagher in 2010, Gallagher et al. Showed that you could be off if you assume the wrong data set within species by up to something like 13.8%. So this is something that, there's been a lot of work in forensic entomology on the environmental side. So what diets might impact the rate at which insects development, et cetera. There's been less work on genetics, and this is something that Christine and I both have been interested in. And ultimately, you could boil this down to some things that people should recognize from forensics and human research, which is, can we predict phenotypes? So there are eye color prediction kits already out on the market, right? So essentially, you might be able to get to the same point with the flies, where I can say, hey, I think that this is one of these genotypes, so I should adjust my expectations as far as how long that species should stop developing or take to develop, and correct accordingly. So the other issue here is more related to the precision issue, right? So I mentioned that intrapuparial development is the last half, essentially, of development. And if you dissect that, you can see you start over on the left side here with these kind of balls of fatty cells. And over time, as they progress through development, they look, essentially like a fly by the end. And so if you know a lot about insect development and morphology, you could dissect these puparia and get a smaller window of time. But there's a restricted number of experts that are capable of doing this, and this is not necessarily a super fast process. However, there are molecular changes happening to this process as well, and it may be possible to identify molecular changes more rapidly that are as informative as an expert in insect developmental biology, as far as breaking this, it could be a 5-day process into a 1 or 2 day window of time. And so just as an example, you could see here, you see the eye begins developing in the middle of intrapuparial development. You can see this light peach color in the eye, and through development, by the end, it's got a nice dark eye color. So that's a potential molecule that one could track to estimate age. And so the idea here is, for a number of things that we're going to talk about today, are there molecules I can use to identify smaller windows of developmental time? And I've done a little bit of this with my dissertation work. So this is from Cherone and Ferran. So in the x-axis here, you have when eggs are laid, and 1 is when adults eclose. So you're going from left to right through developmental time. The black bar here is eggs, first instar, second, feeding third, post-feeding third, intrapuparial development. Then you have a bunch of different genes that are changing expression over time. This is standardized CT, which is basically inverse of gene expression across time. So all these genes are changing in relative expression levels and actual expression levels as development progresses. So you can predict how old these insects are without gene expression or with gene expression. And so this would be, on the x-axis again, 0 to 1 is eggs laid, adults eclose, the predicted age, and then the y-axis is the true age, or proportion development completed. And you can see when you have gene expression included, you get a tighter kind of clustering around the x equals y line, which is the predicted equals true line. So you're identifying tighter windows of time in these later stages of development than if you rely just on the morphological condition of the puparium. Basically all you can say it's in the last half of development. So that's the idea here. So one of the things, though, with this project was that I was relying on the handful of genes that we could get our hands on in the days prior to next generation sequencing. So one of the things that we've been doing is taking advantage of next-generation sequencing technologies to develop transcriptomes for blowflies so that we can then find the best markers instead of just likely markers that are available. All right, so the idea here is I want to, along with my colleagues, work out what molecular markers can be used to make more accurate estimates of age based on what we can assume about the amount of development that's happening in a particular genotype. So is this a fast developer or a slow developer by genotype? And another thing that we can do with respect to accuracy is identify markers of conditions that match our assumptions. So if I assume an insect is growing at a high temperature or a low temperature, can I find a marker for that condition that can then tell me if I assume temperature X, this gene should be up or down regulated. All right. In addition, we'd like narrower windows of developmental time. So the idea here being, can I say that a specimen is 50 to 60% of the way through development instead of in the last half? And in all these cases, there's lots of potential markers. What are the best markers for this purpose? So ideally, they would be robust to lots of environmental differences and ideally similar across genotypes. So we do several things to get at this problem, or this set of problems. So one of the things that we did is when Christine came to my lab, one day she walks up to me and says, I want to do a selection experiment. We had done this with Drosophila, where we had worked with some colleagues that had selected for large and small body size, and then were able to use next-generation sequencing to identify hundreds of alleles of genes that were associated with body size in Drosophila. So the idea was we could do this with development time in a forensically important blowfly. While you do this, you can also do some old-school quantitative genetics and get heritability estimates for a trait, and that basically tells us kind of statistically how much genetics matters for our trait of interest. And so we'll look at that in a little bit. In addition, we're doing expression profiling, so the 2012 NIJ grant has 93 transcriptomes worth of developmental gene expression across 4 different temperatures and 7 different developmental time points. And so we can identify potential markers of thermal environments and these markers of developmental progress. So in the selection experiment, what we did was that we had 2 trials of this. So in 2011, Christine went out into Texas and collected flies from College Station, Longview, and Snook. So you're looking at, in the x-axis here, is the amount of time it takes for the blowfly Cochleomia macellaria, which is common in Texas casework, to develop from egg to adult, and then the counts of each of these individuals. And then in 2013, Jonathan Parrott did the same thing. And so you have 2 different collections by 2 different postdocs. We gave up on Snook, and you'll see why in a bit, and switched over to San Marcos in the second trial. But you can see here, College Station tends to develop a little bit faster than Longview, and that different populations have different kind of means and distributions. These distributions have means that are within about a day of one another, so 258 to 285 hours of development usually gets you to adulthood. And their ranges in development are somewhere between 80 and 110 hours. So you're talking, genotypes differ if you collect from different parts of Texas by about a day. They can differ by up to a day across their development at 25 Celsius, and they can vary, 3 to 4 days in development, essentially. So that's the first thing we got from this. Now what you can do is you can take, if you go back here, you can take all of the flies from this side of the distribution and keep breeding them together. And you can do the same thing for this other side of the distribution. So you can breed faster and faster developing individuals and slower developing individuals. And This is a common, animal breeding technique that, we've used for centuries to do things like develop, lineages of cattle that produce lots of milk. So what you're looking at here is this first generation is that set of distributions I showed you on the previous slide. And the x-axis is generations of selection. Y-axis is development time in hours. So we started just shy of 300 hours in the beginning. Dotted lines are selection lines, and we have 3 controls. So these 3 right here. Individuals are just arbitrarily picked each generation and to found the next generation. So there's a little bit of drift over time in these, but in general, they're pretty flat, and you get fairly big selection response. Faster developing individuals are slower. There's one slow selection group. This is snook slow. For some reason, they kind of tapped out at some point and couldn't develop any slower than that. So I think one of the big punchlines from this slide, though, is that most populations have the capacity to evolve essentially a 6-day difference in mean development times between their fast selection regime and their slow selection regime groups. So there's a lot of capacity for variation and development in these insects. I would point out, though, that that's essentially like starting with a wolf, and then, somewhere over here we're ending up with Chihuahuas and Great Danes, right? So I don't expect to find Chihuahuas in the wild, and I don't necessarily expect to find flies that develop this fast at this temperature in the wild. But we can learn some things from this kind of exercise, and we'll talk about that. So another way to look at this, just this is less busy plot, is you can subtract the fast development time in each generation from the slow development time. And you can see basically over generational time the increase in divergence between those genetic groups. And again, here you see snook, and this again shows about 6 days after over 20 generations of selection. And when you look at those, at the end of this kind of experiment, so here's your control groups. So this is essentially very similar to what the original distribution was. And so for San Marcos, for Longview, and College Station, the fast developers are much faster developing in each case than the controls, and the slow developers, again, are much slower. And you can get these very wide distributions on some of the slow developing groups too. So we're actually increasing variance and development time in some cases. All right, so like I said, these are, it's like breeding Chihuahuas and Great Danes from wolves. What do we expect in the wild, which is really, we're interested in wild flies for forensic predictions. So you can use some information from the starting distribution and from the selection response to estimate something about the contribution of genetics to those phenotypes. And so you can calculate what's known as broad sense heritability from the initial distributions and estimate the variance component for a model that predicts phenotype with just knowledge about what genetic group you're assessing. And that gives you an estimate of 0.38 to 0.44, where 0 would mean genetics doesn't contribute to the variation in the phenotype, and 1 means genetics is the only thing that contributes to variation in that phenotype. So broad sense heritability is somewhere between 0.38 and 0.44. So it's less than 0.5 and greater than 0, so it's probably, in a sense, something we should be concerned about. Narrow sense heritability is a little bit more restrictive. This is basically looking at a specific type of genetic variation where the offspring looks like the average of the maternal and paternal phenotype contribution. So in this case, if mom develops for 8 days and dad develops for 6 days, you expect the offspring to develop for 7 days. And this, is a lower number, but it's somewhere between 12 and 28% of development time variation. And these populations could be attributed to genetics. So it's not clear to me which of these numbers might be more interesting in a forensic perspective, but basically this is telling us in these 80 to 110 10-hour ranges of development time in our populations, how many of those hours roughly are due to genetics in those populations? So this gives us a starting point to begin to assess if we need to tack on some time to our estimates of insect age to account for potential mismatches between assumed data sets and our evidentiary flies. So that'll be a less precise estimate, but it will be a more honest estimate of how old we think those flies are. And then ultimately down the line, we think we can get to something like phenotype prediction where you could get more accurate and more precise estimates of age. But I think we're going to have to do some projects on determining if one type of heritability or another is more important for making good predictions in forensics, and I think there's a lot of area of work that could be done starting from this concept of heritability. One other thing that we can do, though, since we have these strains in hand, is we can ask questions about specific genotypes that might be involved. So are there genes that we think might be important? So forensic entomologists use development time and body size to make predictions of how old insects are. And so there's a gene known as target of rapamycin that is a known regulator of both of these phenotypes. So it's a logical kind of candidate gene. And one of the things about target of rapamycin is, like its name implies, there's a drug called rapamycin that inhibits that gene. So you can actually create what's known as a phenocopy, which is basically you knock out the function of the gene by chemical treatment instead of by mutation. And then you can see what happens. So what you're looking at here is representative individuals from an experiment where we took our San Marcos control fast and slow groups. It's a little bit off, but this is basically a rapamycin minus and a rapamycin plus individual. There's rapamycin plus and rapamycin minus, rapamycin plus and rapamycin minus again. So we've got in a control selection regime, an untreated individual. All these flies are the same age and hours and are exposed to the same conditions. So this individual is, a typical size you expect from this kind of age group, and rapamycin-treated individuals are smaller. If you go to our fast selection regime, the rapamycin control group is about the same size as the control selection group that was untreated with rapamycin, but all of our treated individuals died in this trial. They don't always die, but they're small and they seem to be more sensitive to rapamycin than these controls. And if you go over to our slow group, here's our rapamycin control, and here's the treated, and they're pretty much the same size. So the slows are insensitive to rapamycin, and the fast-developing group are sensitive to rapamycin. So we're hypothesizing that there's either an allele of TOR or of a TOR-interacting protein that is changing the sensitivity to rapamycin and would then be correlated with the selection response. And that's one example of how you can begin linking genotype to phenotype to be able to say, oh, I think this particular specimen is a fast developer for its species. And I'm going to hand it over, I think, now to Christine to let her kind of follow up on some next generation techniques.
Thanks, Aaron. So Christine here, and so this image here, Aaron has already talked about, but it's basically taking different populations of flies and selecting for fast and slow. And then what we did is we actually sequenced the individual genomes of these 3 replicates, so biological replicates of different blowfly populations, and then we sequenced, we did pooled sequencing after 26 generations, which was equivalent to about 5 or 6 day differentiation between development time of the fast and slow. And then the idea was to then start looking at, okay, what is it in the genome that has changed between the fast and slow developers, and what is it between those 2 things that is maintained across the 3 biological replicates? So we started off, we took 2 different approaches to doing this. First, we looked to see, all right, what genes in the fruit fly, Drosophila, are associated with development rate regulation, whether it's increasing it or decreasing it depending on what the gene is. And we selected those, and we searched for those in our 9 genomes, which would be the 3 fast genomes, the 3 slow genomes, and then we also sequenced the starting population so we would get an idea of what the baseline or what the natural genetic variation looks like. After we selected those, there are some challenges when you're dealing with genomes of organisms for which there's no reference to, so you have to assemble your own genome. It's fragmented, so you don't always get good alignment. So in the end, we selected 10 different genes from a starting, I think it's 59 genes, looking for good alignment qualities between the different replicates, and looked for what variants were present. So what parts of the genome changed between the fast and the slow genomes. And then we designed primers, and the idea was these were our candidate genes. How do they actually look in a natural population, but we're looking at the ends of the distribution, so that in the natural population, most of the insects all develop within a pretty short time point of each other, but you will always have the fast and the slows within that distribution itself. So we wanted to look into those tails of the distribution and see whether or not our hypothesis held up. But even before doing that, so here's just an example. So these are a couple different genes that are associated. So sorry, let me grab my pointer here. Okay, so here are a couple of different genes that are associated with development. And in our genomes, which is in the green box here, these are changes in allele frequencies between the fast and the slow. So we had, in these particular cases here, they're all bold because the changes in allele frequency was greater than 0.5. We went into the selection strains, so that is the individuals that we selected for fast and slow development. And we did it for, 30 plus generations, and we had the actual flies from every single one of those generations. But we went into additional flies from the 26th generation that were not used in the sequencing experiment. So that allows us to test whether or not these are just some artifacts from sequencing. And turns out that yes, indeed, in some cases, they are some artifacts from sequencing, whereas the changes in allele frequency didn't quite hold up after we sequenced these individual genes in other organisms. But some did. For instance, this RAS gene, there were 2 variants there that still had a significant change in allele frequency in the selection strains. So then the third test that we did is, this is a distribution of complete development time, so that's from egg to adult, of different replicates of wild-caught flies. So one of my students went out to the park. She collected some of these insects, put them in a cage. We had them produce offspring, and then we collected flies at the various times. And what you'll notice is that here are the fast developers, and then you have the tail end of the distribution, which are the slow developers. So the idea was, okay, so let's test these candidate markers in these individual flies. If our hypothesis is correct, as in if the fast developers have these changes or these specific alleles present versus the slow developers, then we can start trying to increase our precision potentially because we'll know where it falls along the distribution. And so that is being shown right here in this table with the wild offspring. And what you'll notice is that it completely fell apart. As in, for these individual genes, there weren't any changes that were observed in our population data set. Now, I will say with the disclaimer that it was a small population data set, and part of this could be attributed to just differences in the actual geographic origins of the flies. We're talking about Indiana flies versus Texas flies. And we know that flies, depending on where they're from, can be quite different. So we undertook a second approach. And what this is dependent on is just a whole genome analysis. So we first do comparisons between the flies from the different replicates. And what you'll notice is I have 2 geographic replicates here. And as Erin alluded to, the snook one was kind of funny. And so we found it also kind of funny when it came to the genome. So we eliminated it just to make sure that we weren't including something, for example, like we have just a working hypothesis that there might have been a massive reorganization of the genome in the Snook selection line, and so we don't want to have to deal with that. But the other two geographic replicates are much more similar to each other. So first you look for variants that differentiate the two of them. And you want to eliminate those because that's just the intra-population or intra changes between the different populations. And then you compare between your fast and your slows. And that helps isolate individual locations in the genome that might be attributed to these, or at least correlated to these changes in development time. And you can apply statistical tests for doing this. And so what we started off with was 645,000 variants that were present in 18,000 sequences. So these are fragments of the genome. And then we applied a false discovery rate of 0.1%, and that filtered the number of variants in our data set to just a little over 7,000 variants. So these are 7,000 potential locations that are located in about 700 sequences that are statistically different between the fast and the slow developers. So these are candidate sequences. This is where we get started. And some of these sequences, so from 699, only 219 of them had any sort of annotation. What that means is that it is associated with a known sequence that has a known function. And that just tells us that a lot of this is potentially located in non-coding regions, and that's not unusual at all. That's pretty typical. Now, for our purposes, we don't need something that's in a coding region. We don't need something that is in the gene. We just need something that holds up to differentiate between the fast and the slow. Now, if it is present in a gene and we can explain it, then that's very useful for us. So here's one example of a gene that popped out of this analysis, which was associated with protein dimerization. And what I'm showing here is the insulin receptor. And the insulin receptor is really important in regulating development in insects. And so this is just one example of where we need to do a lot more research, a lot more work into all of these individual locations. We're talking about, almost 8,000 different sequences. If I go back to this right here, 7,290 different locations in the genome that are potential candidates. And now it's just mining this data and trying to understand what the data mean. And so just to briefly conclude this part is that we have found several genetic markers that are statistically significantly different between the fast and the slow genomes of these Texas replicates. And so now it's important for us to try to learn more information about these candidate markers and to test them in additional specimens, not just from Indiana, not just from Texas, but from across the distribution for this particular fly, as well as with my lab having access to so many fly genomes, then we can start looking in different species as well to see whether or not the individual characteristics are present in other species, which we would somewhat expect it to be because all of them are under the same selection pressures in nature. And so we found very specific genes that are statistically different that include protein dimerization, and some are regulated by RNA polymerase, which is really important in development time. And so we've gotten to the point now where we just need to dig into this data set more deeply, narrow down the list of candidate genes some more so that it's much more workable, because 7,000 is still too many, and then be able to go out and test these in various samples. And once we do or can do that, then we can start developing development curves that are specific to these genotypes. So you can have a development curve where at a particular locus or a particular location in the genome, it's this genotype, and then develop a specific development curve for that. And then if you were to discover a maggot on human remains, you would subject it to that genetic test and then have a much more, potentially a much more precise development curve that you can be using. Okay, so now I'm going to turn it over to Erin.
Thank you, Christine. All right, so we're going to move over into gene expression. So we've looked at mostly animal breeding and genotyping, and now we're doing transcriptomics. And again, this is to identify smaller windows of development time in some of our kind of less precise development stages in forensically important blowflies. And so, we can find basically when you screen thousands of genes in the genome, you can find lots of examples like this gene here, where from left to right, you're going through developmental time. So these are feeding third instars, early post-feeding third instars, late post-feeding third instars, early intrapuparial developing individuals, and then over here you have late intrapuparial development. And this is normalized, log normalized gene expression level. And so each of these, as I believe 21 transcriptomes contributes to this, to each of these, yeah, I think there's a large number of transcriptomes that goes into each of these distributions, and you can see that there are definitely time points where in early intrapuparial development, this gene is typically much higher, more highly expressed than in late intrapuparial development, where it's off in all of the transcriptomes that we evaluated. Likewise, for this gene, feeding third instars typically have this gene expressed at much lower levels than the post-feeding third instars. So again here, so this is early intrapuparial development, it's at high level, late intrapuparial development, it's off, feeding in third instars, it's low, and then goes up to high expression level. So, lots of genes in the genome. And so, again, this is the same kind of plot for a bunch of different genes, and you can see all sorts of combinations of gene expression levels that can tell you information about each of these points in development that we're interested in. And so what you can do is you can take all the, the thousands of genes that are in our transcriptome, and you can use machine learning methods for these wild-type flies that are all from College Station. We had about 84 transcriptomes that were from the wild-type College Station. We had 9 transcriptomes from the Longview selection experiment population that did not contribute to this analysis. But what you can do is, with this analysis, you can predict the proportion of immature development completed, and this model thinks for this data set it can explain 93% of the variance in developmental progress, and then you can predict development states. So that's just, if I think it's a feeding third instar, is it predicted as a feeding third instar? And it's about 93% effective as well, where 6 of our 84 were misclassified. So these are things like this early post-feeding third individuals that are called as feeding third instars or late post-feeding third instars. But by and large, if we're calling them correctly on a genome-wide level. So with transcriptomics, you can definitely predict smaller developmental windows of time in our problematic developmental stages that are less precise than some of those earlier developmental stages. So another way to look at this kind of data is to cluster samples based on whether or not you can cluster samples based on the expression levels of all differentially expressed genes. And what you can do then is say, look at the principal component. So this is a multi-dimensional representation of our expression information, and this is the first dimension and the second dimension that explain the most variation in our data. So principal component 1 explains 29% of variation in developmental gene expression, and principal component 2 explains 20% of it. You can see we basically cluster intrapuparial development over here, larval development over here. Interestingly, the late post-feeding third instars is kind of greenish yellow color. They start bleeding over towards the intrapuparial samples. And then, like I mentioned, we have 3 Longview slow selected flies, 3 Longview control flies, and 3 Longview fast flies from our second trial of our selection experiment. And so we collected all of these within 24 hours of forming a puparium. So they should cluster with these green early intrapuparial samples right here. And you can see two of the controls do, and then there's this other one that's kind of between the late post-feedings and the early intrapuparial. So we don't know if there's some samples that were maybe very, very recently pupariated, but two of the three of them cluster right where you would expect. The fast-developing individuals, even though they were sampled right after pupariation, are looking more like mid-pupal individuals. And the slow developers, they formed a puparium, but by gene expression, they cluster with larvae. So while they've formed the external structure that looks like a puparium, there's something developmentally that hasn't gotten a hint yet and is leaving a signal behind that makes these individuals look like they're still larvae by gene expression. So there's a lot of remodeling that happens at that point in development, tissues that need to be broken down. So the failure to break down some of these tissues is probably what's contributing to that. All right. And then I mentioned, in addition, you can do things like predict environments. So we looked across our different temperatures. So so it's 12 transcriptomes per developmental time point and then 21 transcriptomes per temperature. So many of our genes had a pattern where at 20 Celsius and 30 Celsius they were upregulated and they were off at 25. And in this condition, which is fluctuating between 30 and 20, which should look a lot like 25. And then we had this one gene that has a different pattern where these are all consistent thermal regimes. They're always exposed to the same temperature, and this group is being exposed to fluctuating temperatures. And so this is a condition that comes up in forensics and may or may not be important in certain forensic entomological analyses, where some species seem to be sensitive to fluctuation in temperature as far as their developmental response. So assuming a constant versus a fluctuating temperature might be important, and this is a potential marker of fluctuating gene expression levels. Okay. Now, I'm going to shift gears. That was the 2012 NIJ grant that we talked about, and that was with mRNA. So mRNA is useful because we know a lot about it, and it's easy to analyze in some ways, but it's not an ideal forensic molecule. And just to give you an idea, when we work with these flies, you flash freeze your individuals and store them in minus 80 as fast as possible in a minus 80 freezer. So you're keeping these specimens very cold to prevent degradation. And no forensic scientist expects samples to be that carefully handled. And so we need ultimately more robust markers. So in this next grant, the 2015 grant, we pursued two different avenues. So one was to look at expression levels of microRNAs over development. And in this case, microRNAs are short and hairpin structured, so they have some double-stranded components to them that whatever it is about these molecules, they seem to persist better in the environment, and there are a number of forensic publications indicating that these may be more useful markers for forensic biology purposes. So we're interested in developmental changes in microRNAs. And you can see here we've got a whole set of microRNAs. So these are third instars, this first 9 set of transcriptomes, and then the last set are intrapuparial individuals. So again, here you can see left to right through development. And the darker blue samples are upregulated. So you start with things that are high in feeding third instars and drop down to lower levels in post-feeding third instars, and then you see upregulation early in pupation and later in intrapuparial development. So again, same marker situation as the mRNAs. And you can see here you have two principal components that explain 27 and 15% of the variation in the data. And you can, in our limited amount of sampling in these experiments, you can see that there are differences between each of these stages, and you can cluster each group individually. Now, another kind of molecular marker that relates to the mRNA but is much more stable and likely to be found is protein, right? So mRNAs, through the central dogma of biology, if you guys remember back to your biology coursework, are going to ultimately be producing proteins, right? So a gene transcribes an mRNA, which translates to protein. So you can look at protein levels as well. So what we have here is this is that first gene I showed you in the beginning of gene expression, where it peaks in expression in early intrapuparial development and then drops off till it's pretty much off in late intrapuparial development. So that's the mRNA profile. And here's a Western blot of the same thing. So here's the early intrapuparial developing individuals that tie to basically this box. And you get a decrease over time in expression of those genes. And that correlates basically to this kind of structural change inside the puparium. So that's basically what we've been up to. So the idea here is you can account for genetics in insect age estimates. I think the first thing that we could do is be identifying heritability estimates, which should be noted are genotype and environment specific, but we should begin investigating if we can use that to account for the fact that we don't know the genotype of our flies. We'll be less precise, but we'll be more honest, more likely to capture the true age of a fly whose age we're predicting. And in the future, we might be able to develop these genetic tests, something like the IrisPlex and other forensic phenotype prediction kits that could be used to predict if you have, say, a fast developer or a slow developer in the future. In that case, you would get more accurate and more precise estimates. Now we can use mRNA, microRNA, or protein to make more precise estimates. Some of these are more resilient to the typical casework conditions than others. I think what might be exciting is something like a pregnancy-style test kit where you just go to the scene, you pick up a maggot or a pupa, you squish it, you expose it to the test kit, and you get certain bands that light up and tell you what age that individual is. And another thing that would be useful is some of the markers we're finding might help us identify markers of conditions that we're assuming or that we're unaware of that might tell us something about whether or not our assumptions are valid in our analyses. So I showed you an example here of fluctuating versus stable temperatures. And at that, I'll hand it over to Christine and John.
All right, here I am. I'm back again. So this story, this collaboration came to be because, well, one, physically, John and I are in the same building, so that's kind of nice that we get the opportunities to collaborate. And two, we had this question about how can we age pupae? It's this stage of blowfly that's very difficult to age because morphologically, possibly you can do it if you're really highly skilled, but it's difficult. And Aaron just talked about trying to do it using other markers, genetic markers, to try to narrow down the time that an individual is during this transition period of metamorphosis. But John, being the chemist that he is, and the ability to differentiate between chemical compounds in a very sensitive manner, we thought, hey, maybe we can make this work. And so the genesis was that if you can determine where along the age timeline that the pupa is in, then you can use that to estimate your minimum postmortem interval. And that Aaron talked quite a bit about that, so I'm not going to spend any time too much on that. And so we were looking for an accurate method to determine the age of a pupa. And other folks out there in the research space had done some research in this field. So I'll skip that. And so we wanted to determine what it was that what could potentially complicate using this method? Essentially, is what we wanted to know. So chemical profiles have been used in lots of different insects to address lots of different conditions. And when they were used in blowflies, it looked really good, actually. So we wanted to know, okay, how can we perturb this system and how will it affect the chemical profile analyses that you do? So we wanted to determine if chemical profiles could just, A, differentiate between species, but specifically differentiate between closely related species. So things that would that are genetically very similar to start off with, so we would expect their chemical profiles to be similar. We wanted to know whether or not the length of time in colony affected the chemical profile. And the reason we wanted to know that is typically when you generate reference data sets, you're working with flies that have been in colony for quite some time. And that might not be representative of the true variation that's present. So we wanted to actually test that. And then finally, we wanted to determine how specific abiotic factors altered the chemical profiles of these pupae. And that's things like temperature and humidity, and even the source of food that the maggot was consuming, because all of that will play into these chemical profiles. So the purpose of this grant was just to test those very specific objectives. So some of the previous work has been done in this area, has used headspace SPEME, and I'm not going to explain that. I'm going to throw it over to John here in a second to explain it. But what they did is they ended up finding some of the prevalent molecules that are present. And so that was our starting point. So I'm going to throw it to John now, and he'll be able to talk about the chemistry side of things.
Okay, so good afternoon, everybody. This is John, and like Dr. Picard said, she came to us originally with this idea. Per my bio, you can tell that my lab is largely involved with arson and explosives, as well as controlled substances analysis. But this analysis actually was perfect in terms of the type of equipment we've got. And it was also good in that we're using equipment that is generally available. So the primary method here is gas chromatography mass spectrometry, which is sort of the go-to gold standard instrument in forensic chemistry. So it's used routinely for looking at controlled substances or indictable liquid residues and so on. So almost every forensic science laboratory is apt to have one of these systems. So the first method that we looked at is the one that's on the current slide. And so this looked pretty easy to us, in that you could use SPEME to detect volatile compounds in the headspace above a sample. So simply put the pupa into a headspace vial, seal it up, let it sit, and then put a SPEME fiber in, which is a very thin fiber with an absorbent coating on it. The compounds would stick to the fiber, you desorb those into a GCMS, and you see them. And so this reference was really useful for that. So the short answer, though, is, and this happens every once in a while, we tried to do this, and it did not work. This, of course, was after we got the grant, and the name of our grant is Headspace Analysis. It has that in there. But we realized pretty quickly that the problem with this approach is that wherever the pupa is being stored, like the environment it is in, it will pick up volatile compounds from that environment. So in the case of the colonies in Doug Picard's lab, there's sawdust there as a substrate. And so we looked at the headspace analysis, and we're seeing all these weird compounds, wondering how they're related to flies, and then finally realizing they're not related to flies at all. They're actually compounds that you see in pine wood. And so they were all, sticking to the pupa and essentially drowning out any signal that we could get from any other compound. So we gave it a good college try. We didn't just give up immediately, but it became clear that there was really no easy way to raise the pupa and then rinse this material off of the pupa and get the rest. We tried different substrates other than pine shavings, and essentially it didn't really give any satisfactory results. So we decided to change our approach. So the approach that we chose here is solvent extraction, and that's as simple as taking the pupa, putting it into an organic solvent, letting it sit there for a while, removing the solvent, and then analyzing that for any kind of compounds that could have been extracted off the pupa. And this has been done before. So there's lots of literature out there where people have extracted insects and flies and pupae and looked at the type of material that's in them. And so the kind of compounds that you would see I've listed here. So like if you've ever heard the term cuticular hydrocarbon or something like that, that's a wax-like compound. You also see free fatty acids, alcohols, et cetera, et cetera. So there's a whole suite of different compounds that can be extracted. And so then the goal was, well, can we get these compounds identified and see if there are any trends between species, obviously between age, and so on. All right, so with Duff Picard's guidance, we had 4 species that we looked at. So that was CMAC, Formia, L. Siri, and L. Cup. And so the fly room that Duff Picard has was set up to segregate the species, but allowed them to be reared in the same environment. So same light dark cycle, same diet, same temperature, same relative humidity, and so on. So we basically could isolate all those different variables so that the only variable that we wanted to test, at least initially, was species. So can we tell them apart in the pupal stage based on the chemical analysis? Then we went on with Formia and did a series of experiments on abiotic factors. And so those are listed there in terms of how those could actually affect the results. Because we realize that we may be using technology, GCMS is broadly available, but if we don't assess how abiotic factors influence the results, it's not going to leap or make the gap between research and practice. Okay, so our sample collection on the left here is a table just outlining the different variables. So we had generations ranging from wild caught all the way up to the 10th generation. The temperature is listed, humidity, etc., and the number of samples. The right-hand table is for the Formia experiments. So in this case, the values that we chose for different temperatures, humidities, light dark cycles, and so on. Now, the method that we chose, this is a method that we've grown in my lab as an alternative to liquid injection. Typical GCMS, you take the extract, you take a microliter of that, you inject it into the instrument, and then you get a result. We use something we call total vaporization solid phase microextraction. So this is the same kind of technique that was used in that initial reference about looking at volatiles that are coming off of a pupae. But in this case, we actually use the organic extract, but we're able to use relatively large amounts. So normally when you inject, I mentioned you inject about a microliter. Here we injected 300 microliters. And we did that by doing this two-step process. First, you have the organic extract down here. There's your spiny fiber right there, and you can see those little arrows denoting that there's an equilibrium between the liquid and the headspace and the headspace and the fiber. But if you have a relatively low volume of liquid and you heat it, eventually the whole liquid vaporizes. Hence the term total vaporization. So when that completely vaporizes, then the equilibrium is directly between the analyte and the fiber, and you gain a significant amount of sensitivity by doing that. And so we've used this approach with a lot of different things, but we also chose to use it here. Okay, so this is sort of a graphic just showing that basic approach, that we know the pupae develop into adult flies, but we're exploring this space oops, there's my pointer. Exploring this space and then doing the solvent extraction and then TV steamy. Something that I did leave out is that the kind of compounds that we ultimately saw, we chose to derivatize them. This is something that chemists do all the time with GCMS. And so that basically means is you react your sample with a reagent that transforms your analytes or compounds into a form that's more amenable to GCMS. So we saw lots and lots of free fatty acids. And so you can analyze those by GCMS, but the performance is much improved when you derivatize them. So this is also something that drug chemists will use occasionally, so you can do derivatizations of amphetamine and methamphetamine to make them go through a GC in a more ideal way, so you get better resolution and overall things are better. All right, so here's a partial list. This is one of our first results that we got, and so this is the chromatogram of Euphoria pupae. And in GCMS, of course, each one of these peaks represents a separate chemical compound, and then the mass spectrometer is there to identify the compounds. And we immediately realized that we're looking at a lot of fatty acids. Those essentially dominate this chromatogram. So here's a list of the different acids. This notation, by the way, 14 in this case means there's 14 carbons. It's a 14 carbon chain in this acid, and then 0 means there's no double bonds. So this is a saturated fatty acid. If you have a 1 like this for palmitoleic, that's an unsaturated fatty acid, so 16 carbons with 1 double bond. So this is just a convenient notation, and you'll see these markings as we go along. But this was an interesting phenomena because as a postdoc at NIST, we were doing research on lipids from skin and hair. And so, and it's the same kind of compound. So they're in different ratios, but essentially what we're doing here is a lipid analysis or a lipid profile of the pupae. All right, so just to prove to you that we're doing the right idea, here's a comparison on the left. This is the response of a fatty acid using either a normal injection, this is the traditional way to do it, or using a TV-Steny approach, so you see a dramatic change in terms of sensitivity. This is the same extract from the same pupae. We split it, so one portion was shot as a liquid, and that's way down here in the blue. The other was analyzed using TV-Steny, and that's way up here in the red. So we were able to be pretty satisfied that we're doing more sensitive work than normal. And then we get into some results. So what you're looking at here is a table from our publication, and it's showing the normalized peak areas for the first generation of a number of species, the 4 species. And so in this figure, black means there was nothing detected, so ND means not detected. Bright red means that there's very high levels of that compound found in the sample. Okay, and so this list of 26 compounds is what we use as the basis for the classification algorithm, and we sort of, as we looked at a number of different samples, said, okay, it looks like we've got these 26 key compounds that could be useful. So some other things to point out, you'll notice that in some cases, all species exhibit high levels of those compounds. So in this case, I'm looking at linoleic acid and oleic acid. But you can also see that some compounds tend to be associated with certain species. Okay, so if you look up here, let me explain this. These are waxes, so if you've heard of cuticular hydrocarbons, these are the large wax compounds. So this particular compound, we're calling it alkane E. The reason why we do that is that we know it's not a straight chain alkane, it's a branched alkane, and those are very difficult to identify based simply on their retention time and mass spectrum. But with this one, you can see that the L. cup species has relatively large amounts of that material, but the others do not. And so just by looking at this, we weren't going to use this as the sole basis of discriminating species, but it was encouraging because you would expect if there's differences between species, then we need to try to tease those out, as opposed to all the compounds being present in everything. All right, so the main tool we used for this is discriminant analysis. We've done a lot of chemometric work in our group, and so this essentially are multivariate statistical methods that can take really complex data sets, like this one, where we have 26 different variables associated with every observation, every sample. So we're talking hundreds of samples characterized with 26 different compounds. So it's a very complex data set. And essentially what discriminant analysis does is it will be trainable. You can tell it in advance what groups the observations fall into. So if you know the species in advance, you tell it that, and it trains itself, and it tries its best to maximize inter-species distance and minimize intra-species distance. So this is called a supervised technique, meaning that you are setting this up to discriminate. So if you were to use this without discretion, it will actually start to try to discriminate between things that actually are identical. So you need to have some sort of prior knowledge going into this. And so then the results, though, will tell you if your prior classification holds up, okay, so whether it's actually reliable. So it does this by calculating another set of coordinate systems. So they're called canonical variates, and they're linear combinations of the original variables. And then it arranges those or aligns those with the different groups to try to, like I said, try to maximize differentiation. Now, to check to see if this is valid, you do a procedure called cross-validation, and you also can test the system with unknowns, additional data. But cross-validation basically means that it will look at the entire data set, it will remove a single point from that data set, recalculate the entire training model, and then take that set or that data point and reclassify it and tell you where it ends up. So if things are working well, your data points, when they are removed from the system, should have little to no effect on the discrimination, and when they're classified again, they should get classified right back where they were. So they should have the same class membership. All right, so the graphs you get from discriminant analysis look like this, and they're very similar to the PCA results that Aaron was discussing earlier, where it's a two-dimensional plot, and every point on that plot is a chromatogram. So each one of these represents a single observation. And so in this case, the system is color-coded. So there's 4 different colors for the 4 different species, and they are aligned on this two-dimensional space, and it shows you to what extent they're similar or dissimilar. And these ellipses that you see here is the 95% confidence limit. Okay, so this is exactly analogous to when we do statistical testing of means. So in this case, if you have 2 groups where the confidence intervals overlap, so in this case we have these 2, so this is, if I can see, CMAC and Formia. Okay, so those are overlapping, and so that would imply that based on this analysis, it's difficult to tell the difference between these 2 species. In contrast, if you have something way over here, so here we have L cup, and so that's differentiable and fairly reliably different from the other 2. Oh, I'm sorry, the other 3. So the difference between the left-hand side and the right-hand side, the right-hand side is the same testing and same basic method, but done on the 10th generation. And so this was interesting because, of course, we realize, and Christine mentioned this, is that you're going to have reference samples, and if we're going to simulate reality here, you might have reference samples of flies that you grow yourself that are have already gone through a number of different generations. Excuse me. But then your casework is going to be over here. So these kinds of flies are the ones that are coming in. So right away we want to see, well, what's the difference? And so just visually, you can hopefully appreciate that this sample groups are slightly more differentiated, so they're less overlapped, okay, after 10 generations. And this seems to make sense to us in that after 10 generations, your species, the one species, is homogenized to a large extent. And so that means that the variation within a particular species after 10 generations is relatively low compared to the variation between species, okay? So this means that if you have a classification algorithm like this, you're going to get very accurate results for both, but your results are going to be slightly more accurate for samples that have been allowed to procreate and homogenize. All right, so here's an example of what this looks like when you're testing accuracy using discriminant analysis. So this is the test on the 10th generation. And so in this case, the training set is all the 10th generation, and we're testing it against itself. So we do take the data out. We do this leave one out cross-validation maneuver, and then test it. But here it looks fantastic. So this cross or confusion matrix says that 53 samples of CMAC were classified appropriately, but one of them was classified wrong or incorrectly, and it's classified as a Formia sample. And so anything that's off-axis like this is a mistake. This is an error, and so you would hope that the number of errors is relatively low. And in this case, it's very, very low. So the overall accuracy is almost 98%. Now, this graphic is talking about the matchup between the first generation and the 10th. Okay, and so in this case, overall accuracy actually does go down. Okay, so you still have a lot of samples lining up along the diagonal, but if you look at the percent correct for each one of these, I've actually forgotten the overall percentage, but it's less than the one before. But you also notice, in particular, Formia has got a relatively low percentage of accuracy. So the problem there is that Formia that are in their F1 generation are sometimes classified correctly, but they're often confused with CMAC and L-Series after 10 generations. So this was an important experiment to show that there's going to be some amount of mismatch between flies that are captured from crime scenes and if you have a reference data set. Okay, and so there's no easy answer here, but maybe we can talk about this afterwards. But this shows you that this actually does tend to suffer a bit. Now, age determination. So I'm showing you results now for Formia, and we started pretty simply. If you just look at the different compounds and sort them and look at the change in that particular compound with respect to age, you can determine correlation coefficients. So to what extent does this compound increase as age increases, or does the compound decrease? And so we found a mixture. Some go down. So for example, mysteric and palmitoleic decrease with age. These three compounds increase with age. Okay, so this is something you could do without a database. So these are all relative and normalized peak areas. So you could actually just look at the normalized peak areas and know that certain compounds are going to increase relative to everybody else, and other compounds are going to decrease. This next column is called sensitivity. So this is if you were to plot the concentration of that particular compound versus time, what the slope would be. So of course you have positive slopes for those that are positively correlated, negative slopes for those that are negatively correlated. And overall it does tell you sensitivity. So for example, the largest number here is this one, 3.74 times 10 to the 6th. And so that means of all these compounds, linoleic acid is the most sensitive. So it increases the most dramatically as age increases. This is linearity, so that basically means this is how well, if you fit the data to a straight line, you are. Obviously ideal would be 1, but some of these we get pretty high linearity, and so that's also encouraging. Now this is the whole view. This is all species at all time points, and it's looking at these 26 compounds, and at the intersection of all these, this is going to be pretty fine print, but you can see these are correlation coefficients. Okay, some of them are in green, meaning they're positively correlated. Some are red, meaning they're negatively correlated. Some of them are just white or gray or whatever. Those basically mean their correlation is pretty weak, and so overall they're not as helpful. But if you take a look at this, some things start to pop out. Like for example, in Formia, there's these two compounds that are very strongly negatively correlated, but that's generally not true with the other species. So you can look at this species by species to see what compounds are more important in terms of at least changing with age. Maybe not making a quantitative estimate of age, but at least changing with age. And so in terms of the compounds, I mentioned these already. These are the waxes that we've seen. These are all free fatty acids, so a whole bunch of different compounds like that. And these are volatile organic compounds, or VOC. So these are the things that you would probably pick up if you just did a headspace experiment. Now, this is near the end of the project, and this is where actually, to be honest, it took me a little bit to understand the importance of this. But Dr. Picard was really clear that, again, we need to know more than just species and age. We need to know what happens if the conditions under which the flies or the pupae are developing change. So one of the first experiments that we did here, and this is with Formia again, is we fed them different diets. So we've got a rat in this case, and then different types of meat in this case. So you can see this is all discriminant analysis again, and the saying "you are what you eat" applies to flies too. So flies are what they eat, meaning that their lipid profiles will differ systematically depending on their diet. This overlap, though, is real, so there's not much difference between low-fat beef and high-fat beef. Okay, so those are largely indistinguishable, but there's a pretty huge difference between those and, say, pupae that were larvae and fed on a rat. So what does this mean practically? This basically means is that if you're gathering samples, it's important to at least have a reasonable justification for them feeding on the same type of material. And so, me as a chemist, I was kind of ignorant of this, but basically, like if you have a maggot that's growing on the liver versus a maggot that's feeding on a different part of the body, to what extent is that different? So here, these of course are easily attainable foods. We didn't test human liver or anything else to see if they'd be similar, but if they're feeding on the same general area of the body, one should expect that there's no difference in their diet. Okay, other abiotic factors. So I use this term, it all falls apart. So discriminant analysis is awesome, but you need to have a reference collection and a sample collection that are fairly similar. So when I first started talking, when you look at these species, if everything's the same, then telling species apart is really easy. However, if you start varying some of these abiotic factors, the accuracy can really suffer. Okay? So I just showed you the effect of diet, and so here's a classification of formia that's been subject to different abiotic factors, and then where it got classified. So in this case, you would hope that all of these observations will be classified as formia, but that's not the case. So, for example, if you have formia fed on different diets, it can have a classification accuracy that's very, very low if you're trying to put it into the same class as formia raised under default conditions. Okay? So if the conditions between your reference data and the actual sample data are different enough, this will tend to fail. Okay? All right, so in terms of potential impact, so obviously research answers some questions and then raises others. So this hopefully sheds some light on how species differ, these particular 4 species, indicated compounds that we know could be useful as indicators of time, but also show the limitations that if you have a reference collection and you have even the same species, if it's been reared under conditions that are very different than the rearing conditions that you use for your reference, then it's going to be very difficult to correctly assign those to the right group. Okay? So in terms of where it stands and what we would recommend, we feel pretty good about species identification. So those differences are pretty systematic and fairly reliable, and it's in a pupae stage, of course, that it's really hard to tell what the species is. Age determinations, that's sort of a work in progress. So we need to examine how those compounds that are correlated with age, to what extent are those, for example, affected by other factors. We didn't do an extensive study of the reliability of age determination with some of the abiotic factors that we did for classification. So I've included references. These are all the chemical analysis papers that I alluded to, and so I welcome you to look those up, and obviously you can contact me or Dr. Picard for additional questions.
All right, thank you so much, John.
That wraps
and
up my
Christine,
portion.
we're going to move into our Q&A now. We do have quite a few questions that came in, and while we are asking those questions, first to our presenters, if you wouldn't mind, if you'd like to give out your contact email address, you can type that there in the chat pod. That way any questions that we do not get to or any follow-up questions, they can throw those out to you. So the first question we'll pose to Aaron, since it came in at the beginning of your talk. Nancy asked, are DNA samples able to be extracted from the various stages of blowfly development?
Sorry about that. Need to unmute. Yes, you can extract DNA from all the stages. I don't know what the if there's a clarification of that question, why you want to do that. Usually it's for species identification. So a lot of people use like cytochrome oxidase sequence. And then obviously what Christine and John were doing is a similar approach. Just how do you identify a species?
All right.
It's usually
the
what
next
people
question we
use
have
DNA
came
for.
in while Christine was giving her part of the presentation, but there's also a follow-up, so we'll just kind of combine them. What if the victim is exposed to drugs? Will those drugs interfere with the fast and slow type fly development? And then somebody else also asked if the person that was deceased was an insulin user, how would those results translate?
Yes, the short answer is that drugs will most certainly affect the development rate of the insects. And there have been studies that have shown the various effects of drugs on the organisms. So you would need to, that would be a little bit of the interpretation portion of your estimates. So you could still do the genotyping and figure out where you fall along the distribution and then adjust accordingly depending on whether or not drugs are present. Now, if the drugs are interacting, then
all right.
That's a different
next question
story,
will.
but I don't know the answer to that one.
Go ahead, Aaron.
If I could follow on that, I'm pretty sure rapamycin is used as a drug for humans in some situations. And so in that specific case, obviously we showed that our slow developers seem to be insensitive and our fast developers seem to be sensitive to rapamycin. So in that case, you'd have different genotypes of flies differentially responding to a drug. And it's also worth pointing out that target of rapamycin and the insulin receptor that Christine was talking about are actually in the same pathway. So again, there's connections here for growth regulation. And
all right.
And
next question we have is for Dr. Goodpaster. Would we need validated development data sets for each fast or slow developer population that we identify, or do we expect to be able to generalize?
Well, I may defer on this one to Christine, but basically, yeah, if you need a reference set, which actually is not clear, which has not been done, is there a difference in the lipids between a fast developer and a slow developer? So if there is, then those could be separated into two different groups, and you would want to classify based on that. Whether there is a genetic question, I guess. So.
Yeah, I guess we haven't done anything on the chemistry side with the selection experiments. So there's sort of two separate experiments. One here is the genetic side, and then one is the chemical side. So I have no idea on the chemistry side whether or not we could differentiate between fast and slow developers.
Well, yeah, and the short answer on the chemistry is that we can if there's a difference. So we're good at seeing small but systematic differences, but I couldn't really speculate on whether the genes that control development time also might control
all right.
lipids.
Next question for Aaron. Will all species of flies have the fast and slow development types?
I'll take this in kind of two pieces. So all species of flies, as far as I know, have some sort of distribution of development times. And so they're, just like, all populations of humans have, distributions of height, right? There are shorter individuals and taller individuals in any group of humans you want to pick, and we're all not the same height. But as far as, whether each species has the exact same genotypes that determine that, that's probably not the case. And just to give you an example, so Drosophila melanogaster has been spread from the Old World to a number of different continents, and on each continent they've developed over a few hundred years the same latitudinal cline in body size, and it's been demonstrated that each of these continental groups has done that in different ways. They use different molecular pathways essentially to achieve the same result.
All right. Next question we have, we'll throw to John. Did you test how the olfactory profiles differ or not for pork and human flesh? Yes.
Okay, when you say olfact, you mean olfactory? Is that? Okay, so yeah, the Headspace Steamy work was done with formia, and it was done under pretty standard conditions. So the diet, I think their default diet in the fly room
Use chicken liver.
Chicken liver. So we did not test for different volatiles. Like I said, though, it became clear that the dominant volatiles of a pupa are going to be those of the substrate. And we did even look at sand or vermiculite, other substrates, but essentially we weren't seeing what that previous work saw. So we just didn't pick up those compounds. And so then, moving from Headspace to a solvent extraction is, a little bit of a blunt instrument in that you are extracting everything if you soak it into a solvent. But with a pentane, that's the solvent we used, just soaking it in pentane for an hour and then decanting off the extract was fine. And this relates to maybe another question, but we even tried this with ethanol. So we realized, or I was taught, that insect specimens are often preserved in ethanol. And the kind of cool thing is that the lipids that we're looking at, they go into ethanol too. So, if it's been in ethanol for a day or whatever, we would just take the ethanol that's already in the vial and we derivatize that, and then we could see the same species.
All right.
So,
next question, or the next few questions we have that came in kind of revolve around casework. So I'll throw this one to Aaron first, but I'd definitely like to get everyone's opinion. What do you see as the next steps on the path of getting your research into casework, and what types of partners do you need to work with?
Are we all going to jump in and answer this all at once?
I can start with
go ahead,
the genetic stuff. So it depends on which project you're looking at. I think in the case of the fast or slow developing issue, there's kind of two stages to it, and I think I talked about that a little bit in the talk. We could probably start applying information from the heritability estimates and that kind of information to current data sets. So if we have some empirical data that, genetic variation is X% of total variation, we can then look at our current reference data sets and pick an assumed amount of variation due to genetics and maybe start doing work to figure out how much time we tack onto our estimates to increase the range associated with any particular estimate. So that's a short-term kind of strategy. The long-term is developing some sort of phenotype prediction kit, which requires, everything that goes into that, which is more analyses of more groups, seeing how generalizable the results are, how many SNPs you need or other genetic variants you need to accurately predict the phenotype, et cetera. That's probably a much longer-term project. Although I will say, somebody recently published on being able to predict human height to within an inch, at least within their data set. So it's possible with enough samples and with enough genotypes and phenotypes, you could get there for the flies as well. With the transcriptome stuff, again, it depends on what you want to do, but I think probably one of the more interesting things is maybe leveraging the mRNA data to identify proteins we want to target and get something like a pregnancy kit style test for pupae and larvae of different ages. And that would require, partners that would develop those sorts of kits.
Yes, I'll echo what Aaron just said. We're a long way, I think, from truly implementing this in the way that it should be used. Currently forensic entomological estimates are being done and like Erin said, they might not be real precise. And so our goal is to try to just increase this precision, and that's just going to take a lot more research, unfortunately. And that is because we need to understand, at a very basic level, what is generating this variation that we're seeing.
I would say, based on the chemical analysis piece, that we have a methodology that works, and so it actually would be pretty interesting to receive exemplars. And like I said, this could be, the actual puparium, or it could be an ethanol preserve sample, or even just the ethanol that was on top of a preserve sample. The problem is that you always need those reference samples and able to enable us to make comparisons. So right now, our reference set is based on frozen pupae. So it works for those kinds of samples, but I think that if we wanted to make this leap into more practical aspects, if the default or the reality is that we're looking at things preserved in ethanol, we're going to have to build a reference collection from ethanol extracts, for example. But that's not impossible, and it's not tremendously difficult. It's just a matter of getting samples like that with known pedigrees.
Next question we have came through while Erin was presenting, but again, this would be a good question for the group. Are these types of tests something that a non-geneticist, forensic entomologist, or CSI would be able to do? And wouldn't you need a lot of specialized equipment? And if so, how do you feel the best way to start bridging the gap between law enforcement and casework to your research?
I would say that, probably somebody in the drug chemistry lab could run these samples if they know what the assay is. And somebody, depending on what the test is, but somebody in the biology section of a lab could be doing the DNA side of things. But unless the whole sort of pregnancy test kit type of test is developed, it is going to require somewhat specialized skill and equipment.
So for microRNAs, if they turn out to be useful in casework stored samples like the ethanol stored samples that Christine and John talked about, there are kits available through like QIAGEN to do qPCR targeting certain loci. So, a lot of labs do have the capacity to do qPCR, so it might be possible, again, for a biology lab to do that. I don't know if the cost is justified in all cases. I think something like the pregnancy kit would be interesting and useful, and probably could be developed in a way that's fairly intuitive for somebody that's even out in the field. There's some long-term things that I think are also interesting, is that at the moment we don't have the capacity to do this, but it's clear that we should be able to at some point sequence large amounts of DNA on site and large amounts of RNA on site, and potentially even protein peptide sequences on site, which means it may be possible, depending on the computational algorithms that people use, to squish up a sample on site and run it through some of these newer technologies. But again, that's down the line as these technologies mature.
All right. Next question we have, we'll throw to John. How would these specimens need to be prepared for TV SPME analysis? Also, this would be really useful for species ID of pupal exuvia. Would this be possible based on this data, and might it be complicated by age of specimens?
Okay, well, these are all great questions. In terms of preparation, like I mentioned earlier, we were dealing with pupae that were frozen immediately upon gathering. Our extraction procedure, though, was very simple, where we just extracted the compound with pentane. This is the same solvent that fire debris chemists use to extract inedible liquid residues off of things. And we did explore, although this didn't make it into our publication, but we did explore this idea of if you have a pupae in ethanol and it's being preserved there, can we take the ethanol and actually get results? And the answer is yes, we can. So the same kind of lipids that we saw in these other extracts will also be in ethanol. And so we would just take the ethanol and process it. So
you would need them to be individually packaged. So
yeah.
you don't want to have a whole bunch of pupae all in one container in ethanol because then you can't analyze that ethanol.
Correct.
Yeah.
Yeah. Then in terms of the other materials, like an empty pupil case, we have not tried that, but I would be pretty optimistic because that outer shell is essentially what we're extracting. Like if you leave a pupa, admittedly, like if you leave it in pentane for like 3 days, it will get pretty mushy and start to dissolve. But when you're just doing it on a short term for most of our results, you can get the pupa back. It doesn't change any. It's just been slightly defatted, if you will. But yeah, that would be something we would have to validate. We'd have to compare intact pupae to the remains of that. But I'm pretty optimistic that that's where most of our lipids lie. Anyway.
Next question we have is in regards to freezing the samples. How would you suggest the best way to collect and freeze those samples at the crime scene? Really for anyone. It came in through Dr. Turone's presentation, but I've heard everybody speak of making sure that they freeze the samples. So anyone and everyone is free to answer.
Well, I'll take from the DNA side, just ethanol.
So for DNA, ethanol is fine. For the mRNA, it's really sensitive unless you can get like specimens that are live and immediately feed something into a sequencer. You want it frozen, and that's not practical for crime labs or medical examiners' offices. So the microRNAs, there's some indication that those are things that might come out of, say, the ethanol stored samples. We have a section in our grant that we haven't finished yet that would get at that for these flies specifically. And then the proteins, it's depending on what assay you decide to go with, you should be able to get information from, for instance, the ethanol stored samples again, or just squishing a sample on site and as soon as you arrive at the scene. So just depends on which test you want to do and what you want to get out of the analysis. And one point I would make with the mRNA analyses is that's useful for then going back and picking what types of proteins you want to target for like a Western blot.
Okay, just a couple of follow-ups. One was asking if dry ice is an option for freezing, or could putting the sample in formalin for testing work?
Dry ice would work fine for most of the genetic stuff, and I assume for the chemical stuff it shouldn't be expensive. Formaldehyde is not recommended.
The next question we have came from David asking, this came in through Aaron's presentation, is this research and information difficult to convey to a jury? Have they been asked what the error rate or UOM is?
So at the moment, we're not implementing this in investigations yet. There are lots of colleagues that do forensic entomology analyses based on the morphology. And like I said, the thing that I think is most immediately applicable is working information from the heritability estimates just into the statistics that people report when they give a range of, I think this specimen is X to Y days or hours old, there's a percentage that you could add on to that to account for the fact that your genotype might not line up with the reference data set. And that, I think it's pretty clear that uncertainty is something that's expected to be conveyed to juries regardless nowadays. So that just seems in line with what's already expected.
I would add that since we're using GCMS, and that's a very common analytical tool, that gets explained to juries every day, when people are testifying about drug analysis or anything else using that instrument. The uncertainty or error rate, you can get at that using discriminant analysis. Like, it gives you this accuracy, and so 1 minus accuracy is error rate. And so you can articulate what the error rate has been for reference collections. So you could give examples. It'd be hard to calculate a global error rate for the technique. I don't know if we're at that point where that is doable, but you could at least express its overall accuracy.
All right. Next question, Athena asked, Aaron, during your talk, so how would we know what type of flies that we are looking at, whether they're fast or slow development? She's referencing the ones on the body that are found on the body because you had mentioned they were offsprings differ.
Mm-hmm. Yeah, so this is a little bit, the inspiration for why we do these kinds of research projects is that, at the moment there's just assumed data sets where somebody measured how these insects develop, and it's with a restricted number of genotypes that they did this, and we all just assume that that's the development rate of that insect at whatever temperature you're dealing with, and we don't know at the moment if we're dealing with, in any one instant, a fast or slow developer, and so getting a handle on how much variation there is and what you do with that is really something that we're trying to do. And like I said before, the heritability estimates are kind of a way that you might be able to account for that without knowing your genotype. And then longer term is developing these phenotype prediction methods, which will need a lot of validation, et cetera, before you could implement them. But at the moment, we're beginning to learn about the biology of what makes a slow and a fast developer.
All right. Next question that came in here at the very end during our Q&A. The studies showed that during the first generation populations have almost the same developmental rate, but after 30 generations, they're different. Those 30 generations were generated in lab conditions with a limited number of individuals collected from each population. If this is correct, how would the genetic pool from wild populations be different from the genetic pool in populations that were kept in laboratory colonies?
Absolutely, that's the case. So in part, the heritability estimate comes from the selection response. And so there are methodologies for continually resupplying genetic variation in a selection experiment to maintain genetic diversity, and there's methodologies where you just start with a pool and then restrict it each and every generation thereafter. And some of the statistics in that table on heritability reflect the uncertainty in that heritability estimate based on that fixed population size, essentially. And so we provided a confidence interval on that, accounting for that, the fact that we didn't add genetic variation. But that's absolutely the case, and I think that's a little bit, things like why Christine was pulling Indiana flies to see if the Texas results line up with the Indiana results. It's going to be important. We're going to have a set of genes that ultimately might look like they are important for a phenotype in one group. Some subset of those will be important for all groups. And in a worst-case scenario, no one gene will be important for all groups. But that's something that no one has a handle on, and we're trying to get.
All right.
We're beginning
we
to
do have
start
time for
thinking
just a
about these
couple more
problems.
questions before we do have to go into our closing. Just for information for our audience, we will have both of our slide decks available in PDF form for download during our closing. So please stick around just for a few more minutes to be able to access that. So the next question we have is asking, does hot water fixation kill or affect the analysis?
Not for DNA, and not for chemistry.
For mRNA, absolutely. Not quite sure on microRNA, but probably. And then depending on if you need a folded or unfolded protein for the protein analysis,
and
may or may not
we'll throw our last question out to John for the day. There's a lot of information using DNA barcoding for identification of flies in all developmental stages, and most of the time for most of the species that is a successful and accurate technique. What are the advantages of using your technique for identification instead of DNA barcode identification?
Okay, so in our method, we're using an instrument that is widely available and overall doesn't takes a chemist to use, but doesn't require extensive training. So we're using something that is on the map already. Also, of course, there's age. So the lipids that we're looking at change with age, which is a major focus of the project. So we already know now which ones are correlated or anti-correlated with age, and so that's something that's not available genetically. And recalling an earlier question, chemical analysis can also be applied to empty pupil cases. So we've got to do the legwork there, but I would estimate that the same chemical signatures are present on that material, even if the fly has already gone off somewhere else.
And it's much faster. So the chemical analysis could be done in a few hours. Doing DNA-based identification, you've got PCR, you've got sequencing, you've got sequence analysis, you have phylogenetic analysis. That can take a couple of days, and most forensic labs aren't doing that. So like John said, pretty much every forensic lab has a
All right, thank you all so very much. We are going to go ahead and move into our closing. Obviously, the first thing we would like to do in closing is to thank our subject matter experts, Dr. Aaron Terone, Dr. Christine Picard, and Dr. John Goodpasser for their research and taking the time out of their busy schedules to talk with us today and deliver today's webinar. You've got quite a few comments coming in there as well to all of our presenters, so thank you very much. Also, thank you very much to all of our participants for joining us today. Without your attendance and your support, these webinars are just simply not possible. So thank you for joining us today. We did record today's webinar, and we will be turning it around into our archivals here over the next few days, so you'll be receiving an email when that is ready. You can check back and reference all of the material today, as well as disseminate out to any colleagues that you feel might benefit from today's webinar. We do also have in the middle of your screen there our short customer satisfaction survey. If you all wouldn't mind taking a few moments to click on that word here and go through our feedback survey, we would definitely appreciate it. All of the information that we do collect is vital information that we use to make sure that we are providing you the best webinar experience, and also to allow us to adjust the way that we disseminate any of the content out to you, our forensic community. You will also see again in the bottom left-hand corner of your screen there our file share pod. This does have both PDF versions of today's slide deck, so make sure you download that before you leave. You will see Julie in there in the chat pod plugging some of our Twitter, Facebook, and LinkedIn feeds, as well as our website and our podcasts. So make sure that you check us out on our website and definitely check into those podcasts. We've had over 35,000 views and listens to our podcasts over the last year and a half. We're very excited about that. So definitely check those out. We have new episodes coming up weekly. Again, thank you so much to all of our presenters, and thank you to our subject matter or to our participants, and we hope everyone has a great rest of your afternoon.